Good evening. My name is Jeff Suzer. I'm the Executive Director at the Powerhouse. Appreciate you tuning in tonight for the second in our three-part speaker series, Vaccines, Viruses, and Immunity. We're delighted to be joined tonight by Dr. Mark Mazur. Mark has a PhD in nutritional biochemistry from Colorado State University. Mark spent 16 years with Abbott Nutrition and has been here in Durango since 2003. He served as a biology and chemistry teacher at Durango High School for 5 years and is one of the longest serving board members here at the Powerhouse. Appreciate Mark coming to join us tonight and share his knowledge on scientific research, the body, how it reacts to disease and vaccinations. This is part of a three-part series that the Powerhouse is hosting to try to help parents as they consider whether a COVID-19 vaccine is the right choice for their families. We're doing one more talk next Tuesday featuring Leanne Jolin and Chandler Griffith from San Juan Basin Public Health. Last Tuesday, we were joined by Dr. Cecile Fraley and Dr. Kelly Miller from Pediatric Partners of the Southwest. Last week's talk is posted on our Facebook page and encourage you to spend time there. Dr. Fraley, Dr. Miller really focused on the conversations they've been having with parents, the questions that they're experiencing most commonly in their practice around the safety and efficacy of the vaccine. Mark's going to talk more about the underlying science about our body, our immune system, and the interaction with vaccines. The other two parts of our project, we've been hosting a weekly vaccine clinic. We have 2 free clinics remaining. One will be this Friday from 2 to 6, February 11th, and the following Saturday, February 19th from 11 a.m. to 4 p.m. Again, these are free and administered by San Juan Basin Public Health. And the final part of the project is offering some thanks and incentives to children who've been fully vaccinated. If you have a child who is fully vaccinated, you're entitled to a free annual membership here at the Science Center. If you come into the front desk with a vaccine record, we'll get you signed up for a free membership. Would love to have you join us at the Science Center, and your child will be entered for a drawing for either a free summer camp or a bike. We'll be giving out 3 prizes at the end of the project, and that runs through February 20th. Tonight, we'll be spending some time with Mark sharing that underlying science. We'll end with questions that have come from the community. If you have questions or would like to share things that come up during Mark's talk, you can email us at info@powsci.org, info@powsci.org. Again, Mark, thanks for joining us tonight, and I'll turn it over to you.
Great. Well, good evening, everyone. Tonight, I'm hoping to simplify some of the more complex concepts having to do with how viruses and vaccines work and a little bit about the immune system. So you might be wondering why someone with a PhD in nutrition can talk about this subject. Well, I started learning how to be a research scientist when I started graduate school in '78 at the University of Wyoming, working on my master's in food science. And after I finished my PhD in nutrition and went to work for Abbott, my first clinical study was looking at how ingredients in infant formula could affect the maturation and development of the immune system in infants. And I've been following that research and engaged in that research, looking at immune development in infants and children for the last 20 years or so. So something I make a point of keeping up to date on. So to start off, we should probably do a little bit of immune system basics. So I'll try and keep this fairly simple for you. The innate immune system is what we're born with. That's really our first line of defense. When you think of our defense, it's good to think about first just the protective system. After all, where is it that we're first exposed to pathogens? Well, it's what we touch, what we eat, what we breathe. So that includes antimicrobial factors like enzymes and saliva and tears. Of course, our skin is a major barrier. Our entire GI tract is lined with a protective mucus layer. Acid in the stomach also works to destroy bacteria. The adaptive immune system is poorly developed at birth. Now, when these innate immune cells, they're very non-specific. They attack anything. But as the immune system matures, these immune systems learn how to recognize specific pathogens. And at that point, they're able to develop antibodies and specialized T cells to those specific pathogens so they can attack and kill them. And most importantly, it also creates a memory so that the next time the immune system sees that pathogen, it's really prepared to act very quickly. And I'll be coming back to that concept multiple times throughout the talk. So let's talk about how that actually happens. Here you see a macrophage ingesting a bacteria, and it totally digests that bacteria, and it isolates what we call antigens. And these are structures, mostly protein in nature. And oops, got the wrong button there. There we go. So it isolates those antigens and then attaches them to a molecule that then takes that antigen to the surface of the cell and presents it. At that point, a cell called a helper T cell can bind with that antigen on the macrophage. And how does that helper T cell know to do that? Well, in order to recognize that antigen, the macrophage sends out cytokines, chemical messengers that alerts the helper T cells to come to that macrophage and bind to that antigen. And that activated helper T cell is also then sending out these chemical messengers that coordinate the activity between different cells in the immune system. So in response to those cytokines, the T cell clones itself. It makes lots of copies. And those cytokines that it secretes activates the T cells as well as B cells. And then the T cell and the B cell can communicate directly. They actually attach to one another. And then finally, the B cell then transforms into plasma cells that produce those antibodies. Those cytokines that activate the T cells also cause some of those T cells to become cytotoxic. They're also called natural killer cells. I guess you could say they're natural born killers. And these NK cells will directly kill any human cell that's been infected with a virus or has become cancerous. So if a cell has been infected with the coronavirus, that cytotoxic T cell will destroy that cell. The key to adaptive immunity is really that activated T cells produce memory T cells and activated B cells produce memory B cells. Here you see the basic types of immune cells in the immune system. And what you see in all those little boxes are these cytokines, these chemical messengers. It's how the cells not only communicate with each other, but how they coordinate their attack against a pathogen. You may have heard the term cytokine storm. When some of these cytokines, which are inflammatory in nature, are overproduced, it can cause a generalized inflammation that can be very damaging. So for instance, this cytokine, TNF alpha, is thought to be the cause of rheumatoid arthritis. And the treatments for that are antibodies that bind to that cytokine, thus preventing the RA from expressing itself. Rather than give you a description of each of these cells, I like to show pictures. So think about that sequence I just showed you about producing antibodies. So first, that macrophage, acting as a antigen presenting cell, eats that bacteria, presents that antigen to a T cell where they're communicating directly. Then that T cell communicates directly with a B cell, and then that B cell eventually is able to release antibodies that directly kill the virus. This is what's actually going on inside of a B cell when it's communicating with that T cell. Now, of course, I don't really expect you to read that. I just want you to be impressed with the vast number of little metabolic pathways that are going on inside that B cell while it's preparing to produce those antibodies. Keep in mind that each one of those pathways is probably a research project from a scientist or a group of scientists, a vast amount of work in deciphering those pathways. Here you see a depiction of antibodies actually attacking a coronavirus, attaching to those spike proteins so that it won't be able to bind to human cells. This is a depiction of a cytotoxic T cell attacking a cancer cell. It would be doing the same thing to a cell infected with the coronavirus, attacking it and killing it so it won't be able to allow the virus to spread in the body. Now let's look at how the immune response works. After you're first exposed to a pathogen, you start producing these antibodies, and you want to be able to get to a protective level. And you can see that it takes about 2 weeks before we are able to get to that protective level. And then after that response and the disease is controlled, you see the antibody levels fall to below that protective level. But remember that the adaptive response also produces memory T cells and memory B cells. And because of that, the next time you happen to run into that pathogen, remember antibody levels are below the protective level, so you may get infected. But because of those memory cells, you see that the response is within just a few days. And it's much more rapid, much more vigorous, and much stronger, a much more robust response so that you don't get really sick. A vaccine works in the same way. A vaccine is similar to being the first time you're exposed to a pathogen. It produces antibodies and also those memory cells. And again, eventually the antibody levels are reduced. But then when you're exposed to the actual virus, because of those memory cells, you're able to produce this really robust response that protects you from serious disease or hospitalization or death. So a virus binds to a human cell through some kind of a receptor that it recognizes. Then that virus injects RNA into the cell. Then the viral DNA takes over the cell's synthetic machinery and produces copies of the virus. While this is happening, millions of copies of this virus are produced in the body. A vaccine, it can be either a dead virus that can't really cause the disease, or it can be a part of that virus, such as a viral protein. And the immune system then recognizes these as a foreign antigen, and it is able to make antibodies to that antigen, providing protection. Here we see the picture we've all become fond of, that beautiful coronavirus with all of the red spike proteins covering its surface. And that spike protein is what binds to a receptor. So here's a depiction of that attachment. So here you see the ACE2 receptor. That's the receptor that the coronavirus recognizes. And here you see that spike protein binding to that receptor. Now, to explain how the mRNA vaccines work, I'm gonna have to do a little bit of high school biology for you. We need to go over a little bit of how protein synthesis works. So here's a cell, and in the nucleus of the cell is DNA. The DNA contains the instructions for making any protein that that cell has to make. When the cell gets instructions or needs to make one of those proteins, it takes the instructions for that particular protein and puts it onto a messenger RNA. That mRNA then goes out of the nucleus into the cytoplasm and finds a ribosome, sort of the assembly line for making proteins. Then transfer RNAs bring the amino acids to that ribosome so that it can make the protein. If I can give you an analogy, let's say that that cell is actually a chocolate factory. So the DNA of the chocolate factory will contain the instructions for all the chocolates that that chocolate factory has to make. Then the factory gets an order for truffles. So the recipe for truffle would be the factory's messenger RNA for truffle making. Then the transfer RNA would bring all the ingredients to the assembly line, which would be the ribosome of the factory, and then it can produce the truffles. So in making the Moderna and Pfizer vaccines, they used messenger RNA technology. Now that technology has been in development for over 2 decades. And the real big advance came when scientists determined how to get the messenger RNA into our body without it being destroyed by enzymes that are in the blood. To do that, they used a liposome, a fat bubble, and they put that messenger RNA that contains the instructions for making a spike protein into that liposome. Well, that liposome can bind with the cell membrane and then release that messenger RNA into the cell. It finds a ribosome and then binds to that ribosome so that the ribosome is then able to make those spike proteins. These are then put on the surface of the cell so that an antigen presenting cell, one of the immune cells, can recognize that spike protein and then go through the process of eventually turning on B cells to make antibodies to those spike proteins. The AstraZeneca and J&J use an adenovirus vaccine. In that case, what they use is an adenovirus. It's a virus that is basically unable to reproduce itself. And into that virus, they put viral DNA. It's actually a synthesized DNA, and it contains the instructions for that spike protein. So when that adenovirus binds to a human cell, the cell uses that recombinant DNA to make spike proteins, which then the immune system can make antibodies to. So there are a lot of terms around this use of vaccines. So what is efficacy? Here are the data from the J&J vaccine trial in the US. There were 63 volunteers in the placebo group who got COVID. In the vaccine group, only 18 out of 5,000 got infected. So 72% fewer cases were in the placebo in the vaccine group. That means it has an efficacy of 72%. That's how they come out with that number. For the Moderna mRNA vaccine preventing illness, in the vaccinated group, only 55 people out of 15,000 became infected, whereas 744 out of a little over 15,000 in the unvaccinated group became infected. So that's an efficacy of 93.2%. Now keep in mind that even if you had an efficacy of 95%, that means that 1 out of 20 people would not be protected. No vaccine is 100% effective, just as not every person's immune system is going to be 100% effective in protecting us from different illnesses. What's more important is how efficient it is in preventing serious disease. In the vaccinated group, only 2 out of over 15,000 people had serious disease, while just over 100 in the unvaccinated group of over 15,000 had a serious disease. So that's an effectiveness of 98%. And that's really the important part. When we look at the Pfizer vaccine, the efficacy against infection 3 days after the first infection was about 35%. About a month after the second injection, it rose to about 75%. But 6 months after the second injection, that first series, the efficacy dropped to about 20%. Remember that after the antibody levels first are produced, they fall off. And that's what that really shows. But what's more important is the effectiveness against severe, critical, or fatal disease. So after 3 weeks after that first injection, it was 65% effective in preventing severe disease. After 2 months after the second injection, that rose to about 98%. But look, 6 months after the second injection, that effectiveness against severe, critical, or fatal disease was still at 98%. And that's because even if one is infected, our memory cells kick in and we produce antibodies and killer T cells to go after the infection. So efficacy is the percent reduction in the incidence of the disease, the actual number of people that get the infection. Whereas the vaccine effectiveness really describes the ability to prevent outcomes of interest in the real world, serious disease, hospitalization, or death. Now, there are now a couple of antiviral pills on the market. The monoclonal antibody infusion required going into a hospital for an infusion. These pills are much more useful. You start taking them within 3 to 5 days of becoming infected. They're taken for about 5 days, and they're produced by either Merck or Pfizer. And the percent reduced hospitalization after 30 days was about 30% for the Merck pill, with only 1 death. And for the Pfizer pill, the percent reduction of hospitalization was 85% with no deaths. The challenge with these pills is that because they're new to the market, manufacturing has not yet been able to keep up with the demand given the number of infections in the country. So why do you need to wear a mask if you've been vaccinated? Well, you may have a subclinical infection. In other words, you could be asymptomatic. You don't even know you're infected, but you still may be able to transmit the disease. So again, remember 95% efficacy still means that 1 out of 20 people is unprotected and you can still transmit that disease. A good way to understand protection is looking at what's called the Swiss cheese model for protection. So imagine you had a block of Swiss cheese and you sliced it up into different blocks. And here you have the virus and it's trying to get all the way through to you to cause the infection. Well, the holes don't actually line up. So the more of these slices you can put up, the more challenging it is for the virus to get to you. And these slices can be things like distancing or staying home if you're sick, of course, wearing masks, hand washing, not coughing into a room, but coughing into your arm, not touching your face, avoiding crowds. Testing, of course, is critically important. And Ventilation, air ventilation in rooms or being outdoors. Government support can provide some protection. Then quarantining or being in isolation around and keep you away from infected people. And of course, most important, vaccines. Now, the first vaccine that came out, it was against smallpox. And this was back in the late 1700s. And it was made from smallpox, from cowpox, uh, infection similar to smallpox, but didn't cause serious disease in humans. And at the time, there were all of these images that came out trying to scare people about getting the vaccine. And some of those images of how the vaccines were made and given were pretty frightening. And here we are 200 years later, and we're still getting these messages about all these things that vaccines are supposed to do that are based on nothing factual. There's nothing factual indicating any of these stories are true. No, there are no chips in vaccines. They don't cause infertility. All those have no basis in fact. The first vaccine mandate came from George Washington when he wrote to the, what would be equivalent to the Surgeon General, I guess, ordering him to inoculate all of the army that came through Philadelphia, saying specifically that it's necessary not only to authorize, but seems to require the measure for should the disorder, that would be smallpox, infect the army, we should have more to dread from it than from the sword of the enemy. In fact, a lot of historians credit the use of the smallpox vaccine by Washington to be a major contribution to the success of the Revolutionary Army. When we look at this graphic of deaths per 100,000 residents, you see that the greatest concentration of deaths is in the Southeast, which is also where we find the lowest vaccination rate. That's not a coincidence. People often question how efficient VAS are. There are now at least 11 studies published in medical journals showing the efficacy of wearing masks in preventing the transmission of disease. Masks definitely work if they're, especially when they're properly put on and not pulled down under your nose. And the reason they work is that because the way coronavirus is transmitted is through the air. It's aerosolized. Here is a photograph of someone coughing or sneezing, and you can see this explosion of particles out of that person's mouth. The real challenge is that some of those particles are tiny. They're less than 10 millionths of a meter in diameter. And because they're so tiny, they can remain suspended in the air for hours. And if they contain the coronavirus, they can still cause infection. And that's why ventilation in rooms is so important in preventing the spread of disease. Here you see the different parts of the body where the ACE2 receptor is found. Remember, that's the receptor that the coronavirus binds to. Most predominant in the lungs and the ileum, part of the GI tract, also in lymph nodes, but it's also found in the kidney and the heart. And you can see why there are so many different symptoms in different people, because it depends on where that virus proliferates most. This is a micrograph of human heart muscle, here shown in red, that's infected with the COVID virus, shown in green. You can see that it can be a very nasty heart infection. In fact, there are ACE2 receptors even in the brain. So this virus is very unique in how many different parts of the body it can cause infection. And again, it accounts for the wide range of symptoms that we see in this disease. Now, what about all these variants? Well, these variants are really evolution in action. Consider how long it took the woolly mammoth to evolve into an elephant. Well, assume a mammoth has a gestation like an elephant, about 22 months, and only one offspring. Well, that process took millions of years. But when a virus mutates into a new variant, that replication is only hours long, and it produces millions of offspring. So that evolutionary change is only weeks to days. In reality, there probably have been thousands of new variants over the couple years, but the only ones we see are the ones that are successful. To be successful, that new variant has to find a new host. It has to find an uninfected person, and also has to be viable, of course. And to spread, it generally has to be better than the variants that are already out there. That's why it's so problematic to have so many unvaccinated people, because it provides an opportunity for new variants to spread, to find a new host, and to become established. So the first variant that was of real concern was the Delta variant. That was actually our second wave, the really damaging one. It was first identified in India, and it spread much faster than previous variants, which we saw that led to that huge spike in infections. And it was also much more severe than previous variants, increasing dramatically hospitalizations and death. Now what we're just coming out of, thankfully coming out of, is this huge spike from the Omicron variant that was first identified in South Africa. Now it's spread much faster even than the Delta variant, but severity is much less than the Delta variant. But just because the severity is less, that shouldn't mean you should not be concerned. Over the last 2 weeks, over 2,500 people in the United States have died from the Omicron virus. It may be less severe than the Delta, but that doesn't mean its severity can be ignored. People are dying from it. And you can see that in the daily trends. This is from the CDC. And late October of last year, there were over 486,000 cases, leading to a 7-day average of over 300,000 cases a day. In just over 2 months, by early January, we had over 1 million new cases reported, with a daily average approaching 500,000. This is the Omicron variant. And you can see the dramatic effect that vaccines have when you look at the huge gap in hospitalizations during New York City's Omicron surge. Here in the upper line are the hospitalizations of the unvaccinated. You can see it's many times higher than the number of unvaccinated people, the number of vaccinated people. In other words, the crush that our hospitals are experiencing now, including here in Durango, is really being pushed by unvaccinated people, creating this crisis in our hospitals. It's also seen pretty well in this data from nursing home residents. Nursing home residents are the most vulnerable population. Here you see the cases per 1,000 nursing home residents and those who are unvaccinated. Here are the residents who had the primary series of vaccines. In other words, they got 2 of the Pfizer or 2 of the Moderna or 1 of the J&J. But if they got the booster, it dropped to close to 0. That shows the importance of getting that booster, which not only bumps up antibody levels, but also will increase the number of memory T cells and memory B cells, so we're better able to fight off that infection. Now let's turn our focus to children and 5 to 11-year-olds. Right now, Pfizer is the approved vaccine for those children. Looking at the cumulative cases of COVID in these children, this is the percent of subjects after the first dose, about a 10-fold difference between those who got the vaccine in orange and the placebo in yellow. So that's about 0.25% of the vaccinated children got infected. And after the second dose, that difference remained. So to put some numbers on that, after that second dose, only 3 out of over 1,300 children in the study became infected, where 16 out of just 663 in the placebo group became infected. And a lot of people want to know more about how children react to that vaccine, not just in terms of infection, but the most common response, of course, is pain at the injection site. Here we're looking at the percent of subjects who experience pain. And after the first dose, about 75% of children in the vaccine group experience pain and 30% in the placebo group. Let's face it, getting poked by a needle can be painful, whether it's the vaccine or the placebo. And that difference remained after the second dose. What about more severe adverse events? So any adverse event. And let me take a step back to explain how these data are collected. When a company wants to submit a clinical study to the FDA for its approval, it has to do what's called an intent to treat study. That means that you have to keep track of every subject who is enrolled in the study. That's anyone you intend to treat. And that includes both in the group actually receiving the medication as well as the placebo group. So let's say you enroll a subject in a study and the next day they move to Timbuktu. Well, you enrolled them in the study. That means you intended to treat them. So their data has to be included in the submission to the FDA. Now that makes a lot of noise in the study because you're collecting so much data, but it also ensures that any significant difference you do see can be trusted. So during these studies, every single event is recorded. Every doctor engaged in the study has a clinical research monitor assigned to them who goes to the study site and records and documents any adverse event that's reported by any subject in the study, whether they think it's related to the medication, or in this case, the vaccine, or not. So in the Pfizer study, and this is from the clinical study in the New England Journal of Medicine, and this is percent of subjects, 3% of subjects in the vaccine group reported any adverse event that seemed to be related to the vaccine. And 2% of the placebo group also reported an adverse event they thought that was related to that injection. As far as, and this includes things like pain, fever, fatigue, headache, chills, vomiting, diarrhea, joint muscle pain, use of pain medication. And in terms of severe reaction, it was less than 0.25%, a tiny amount. And even if we look at these severe reactions, this is from the CDC morbidity and mortality report, the end of December of 2021, by that time, 8.7 million 5 to 11-year-olds were vaccinated. There were little over 4,200 reports of adverse events. 97.6% of those adverse events were not serious. The chance of a serious reaction was about 17 thousandths of a percent. This is a tiny amount. And most of those serious, in fact, half of those serious reactions were fever and vomiting, not necessarily life-threatening. When we look at the attitudes of parents of 5 to 11-year-olds towards getting their children vaccinated, this is from the Covet vaccine monitor study. Back in July, about 25% of parents said they would definitely get the vaccine when it was available. About 40% said, well, they were hesitant, they would wait and see what it looked like when they started seeing reports. Another 7% say only if it's required. And about a quarter of parents said, no, they're not going to get their kids vaccinated. It changed somewhat by September, but we see by October, it has changed pretty dramatically. So that now about 73% of parents of 5 to 11-year-olds are either hesitant or reluctant or definitely decided they will not get that vaccine. That means that all of those children are vulnerable to getting infected. And remember, although they have a mild infection as a rule, that doesn't guarantee that they may not have a serious infection. When we look at the percent of cases in the US by age group, people across all ages do get infected. And in that 5 to 11 age group, over 2.7 million children in the US have been infected. And there are 566 cases in 0 to 9-year-olds here in La Plata County alone, and 661 deaths in children in the last 23 months. To give you a perspective on that, before chickenpox had a vaccine, there were about 70 deaths per year from chickenpox. So yes, it is generally a much more mild case, but that doesn't mean that it can't be serious, and although infrequent, may cause death. And vaccines are not new. We've had them for years. Here are the current vaccine recommendations by the academies of the American Academy of Pediatrics, also OBGYN, primary care doctors. You're familiar with most of these and probably have had your children vaccinated. What you don't see up there is smallpox. Why not? Because the smallpox vaccine was so effective that it essentially has eliminated smallpox disease from the world. That's quite a change from back in the late 1700s when it could turn the course of a war from winning to losing. And by the way, the efficacy of that smallpox vaccine was about 95%, similar to what we see with the mRNA vaccines. So when you're thinking about whether or not to get a vaccine, you have to weigh the risks versus the benefits. What are the risks? Well, it could be a mild disease, but it could become severe. It could lead to loss of taste and smell that can be long lasting if a person develops long COVID, in which case, even though you may not be infectious, the symptoms can persist for months and even over a year. And then, of course, in the more severe cases, leading to hospitalization and death. And the benefits, well, these vaccines have proven safe and effective. They significantly reduce infection, hospitalization, and death. And this has been shown in millions of patients all over the world. As for sources of COVID information, probably the most reliable sources are the CDC, or if you're in Durango, the San Juan Basin Public Health. If you're outside of Durango, your county public health also will likely have a website. So if you just Google CDC or your county public health office, you'd be taken to their website, which will have answers to all of your questions. Well, thank you for listening. Hope this was a good talk for you, and I'd be happy to take any questions now for any of you who have any.
Terrific. Just as a reminder, if you do have questions, please send them to info@powsci.org. Mark, I've got a bunch of questions for you. I think maybe starting with, I think one of the things that just jumped out to me, and I just kind of sort of watching my own world, when you talked about efficacy versus effectiveness, I think that really resonated for me. I was vaccinated early on, and I kind of felt bulletproof for a few months. When I read, what was it, 92 or 93% chance of, reducing my likelihood of getting the disease. And as we've looked at the numbers, it just doesn't seem like vaccines are as effective as it looked like they were a year ago. And it feels like the goalposts are moving. Can you talk a bit about that? I'll leave it at that.
Good. So again, efficacy is really a description of, during the clinical trials, how efficacious is the vaccine in preventing getting the illness. But recall from the graph I showed you that eventually, and really what that's based on is how many antibodies you produce so that you can actually block getting the infection. But those antibody levels fall, and eventually they fall below that protective level. And even in a very good vaccine, efficacy 95%, that means 1 in 20 people may still get infected. But if your immune system, or if you're vaccinated, produces those memory cells, then even if you do get infected, your immune system has the ability to protect you. So I personally got the Pfizer vaccine, and I got the Pfizer booster, so I was protected, but I did get a breakthrough case. Yes, you can still get infected. Again, no vaccine is 100% effective in preventing disease. So I had a breakthrough infection. I had a runny nose, coughing, sneezing, a little bit of muscle aches for 3 days, and then it was gone. If I hadn't had the vaccine, it could have been a much more serious infection. Because I was vaccinated and because I have those memory cells, I was able to mount a much more vigorous response, and it was a very mild disease. It wasn't fun, but it was no big deal. It was no more than the annual cold virus. My wife, who has, let's just say, multiple underlying conditions, including taking an immune suppressive drug for rheumatoid arthritis, she also had a breakthrough infection. Now, theoretically, it should have been a lot worse than mine, but her symptoms were even more mild than mine. She was really tired for a few days, and that was about it. So we see a wide range of responses to this virus, both in vaccinated and unvaccinated people. But if you have been fully vaccinated and boosted, you're much more likely to have a very mild case, and you're extremely unlikely to be hospitalized and extremely unlikely to die from this virus. So
And
kind of
Mark, I
get
think one of the things that, yeah, no, and I think one of the uh, things that I appreciate from you in the conversation is I think most of us live in a world of anecdote. I know somebody who was unvaccinated, they got COVID, it was no big deal. Why should I worry about vaccination? Or I know somebody who was vaccinated and they still got COVID. Why would I bother getting vaccinated? Clearly the vaccine doesn't work. And I think what you do a good job of speaking, reminding me of is we think in terms of sample sizes in the tens of thousands, hundreds of thousands, and millions, and that that it's the weight of large numbers that tell us what works and what doesn't.
Yeah, and think about, I mean, when I was a kid, there was no vaccine for measles or chickenpox or whooping cough or mumps. So pretty much everyone got them. And there used to be like measles parties because people thought, well, if I intentionally get my kid infected with measles, then he'll be protected from getting a more severe case. Um, and believe it or not, there have been COVID parties, but thousands of kids died from measles. It was not an innocuous disease. So when the vaccine became available, it has saved thousands and thousands of lives. So yes, getting the disease can offer protection, but does it offer, so that's what people are thinking about when they say they have natural protection. Oh, I got the disease, I have natural protection. Well, if that natural protection comes at the cost of being hospitalized and thrown on a ventilator and possibly dying, it's probably not worth it. And again, if you can get equal protection and long-lasting protection from vaccines, that seems to me to be a much safer alternative.
And Mark, as I hear you say that, I think it's partly when what's the risk of getting vaccinated versus what's the risk of going to a COVID party is that you're comparing. I forget the number that you cited. Was it 17 10,000ths of a percent as a risk level for a serious reaction versus? Can you give me a point of what's that comparison of an unvaccinated person? Again, most people I know who've had COVID, vaccinated or not, have been okay. But how big of a risk is COVID at this point for someone if I'm saying, hey, I'm just going to get COVID, forget vaccinating me, I'm just going to get COVID?
Well, again, 2,500 people a day in this country are dying from being infected with the Omicron virus. And it's been that way for 2 weeks. Even though the number of cases, the daily number of cases has really, really fallen pretty dramatically over the last few weeks, the number of deaths has not fallen. It has continued to be at a very high level. So this is not an innocuous disease. And even though for many people it may seem that way, it certainly doesn't if it's been someone in your family who ends up dying from this disease.
And
We've had over 900,000 people have died from COVID in this country.
so $2,500 deaths a day during this surge. How does that compare to how many people die in a typical day from getting vaccinated?
From getting vaccinated, I'm not aware of any deaths caused by vaccines.
Yeah, well, and so as we started this series, I've been getting emails from people. I got an email that told me that 22,000 people died from getting the COVID vaccine, based on the data, the adverse incident report. Can you talk about that? Again, I got an email that said there were 22,000 plus COVID vaccine related deaths.
Yeah, let me trying to get to a okay, this is from the what's called Vigi Access, which is the World Health Organization reporting system similar to the VAERS. So these systems are set up so that anyone can call in a report of an event that they feel related to the vaccine. So this is what is posted on that website. The information on this website does not reflect any confirmed link between a medicinal product and a side effect. Vigi Access cannot be used to determine the likelihood of a side effect occurring. It cannot be used to compare the safety profiles of different medicinal treatments. The information should not be used in isolation to make any decisions regarding a patient's treatment regimen. So in other words, these reporting systems are not the same as the reporting systems that are used in a clinical trial. Remember, in a clinical trial, clinical research monitors go to every single clinical site that's included in the study, and they record and document every single event that occurs in the study, every event, regardless of whether it's in the vaccinated group or the placebo group. And every single one of those events has to be reported to the FDA. You see this when, if you ever look at say, an insert into a medication, or when you see one of the advertisements on TV for a medication, and you see in the background happy dancing people, and then at the bottom is listing all the possible side effects. The FDA requires, it's the law, that companies report every event that occurs during the study, no matter how rare or whether or not it is proven to be linked to that disease. Now you can look on the insert, it will tell you how many of those events occurred. In other words, how likely that adverse event is to be associated with the medication. But when you hear that plethora of side effects, that's because the law requires that every single thing that's reported is notified. But these public notification bases aren't rigorous data collecting systems where the report is validated by a scientist or anyone determining how we know whether it's actually connected to the event or not.
And yeah, I think how high is that bar? What level of side effect or adverse outcome triggers, the reporting agency say, let's rethink whether this vaccine's safe. Let's put out a warning to the community that you might think twice about trusting the Pfizer vaccine.
Well, so when you look at the approval process, there are several steps. So the FDA has an advisory committee, and they're given all of the data from the clinical study. And as you can imagine, that's volumes of data. When you think about every single event that occurs in a study over the course of many months. And they examine all that data and they have to decide what adverse events can be linked to the vaccine. And if they, so that they have to make a judgment on how effective that vaccine is, first of all, in preventing disease and also in being safe. So the safety of a vaccine or any medication is really, really important. And based on that evaluation, which includes very complex and detailed statistical analysis, they make a recommendation to the FDA. Then the FDA has its own experts who then look at the recommendation from the advisory committee and make a determination whether or not to approve the vaccine. Safety is really the primary concern of these agencies, of these scientists, to make sure that it is safe. And then once a medication is put on the market, there is another process called post-market surveillance. So even after a medication has been approved, people who use that medication are followed, and if something were to pop up, because even a large trial may have thousands of patients, and then once it goes on the market, there are, of course, millions. And then there may be something that's a safety problem that occurs that can only be seen in millions of patients. But let's look at the current vaccine. At this point, we have that data now. Over probably by this time, close to 90 million 5 to 11-year-olds have been vaccinated, and we just haven't seen those reports of any serious adverse events.
And just the other set of questions I've gotten from people is around financial interest. Why should we trust Pfizer when Pfizer stands to make, money in the millions, if not billions, by marketing a vaccine? Why should we trust their studies? Because aren't they compromised by, profit motive and self-interest?
Well, I mean, obviously, these companies have a profit motive. They intend to make money from these developments, but that doesn't mean that they're not held to extremely rigid standards by the FDA. So I worked for a pharmaceutical company for 16 years, and we had very rigid rules about how to conduct these clinical studies. I remember in one of my first studies, I can't remember exactly what the study was. We saw a difference in growth of infants on one of the arms of the study. And when I looked at the data, there were a couple of subjects in one arm of the study that were real outliers. They were clearly different than the rest of the group. And I saw that, well, if you took those outliers away, then the difference would disappear. So I'm sitting with the statistician. I said, well, why don't we just throw those outliers away and then we wouldn't see a difference? He said, Mark, we can't do that. That's against the rules. You have to include all of the subjects, all of the data. So there are internal rules and external rules. It wouldn't behoove the company to produce, I mean, if you produced a medication that ended up having serious effects that led to serious disease or death, what would that do to the company? It would destroy their reputation. They would no longer be a profitable company. They would stop making money. Everyone makes money from being good at what they're doing. Doctors get paid for treating disease. Construction workers get paid by building good buildings. When you're wanting to build a new house, who do you go to? You look to contractors who have a really good record, and they may be a little more expensive, but they do a better job. So why should it be different with pharmaceutical companies wanting to get paid for producing medications that save lives?
Can you talk about emergency authorization? Again, concerns are, I get a couple of concerns I hear from some of those hesitant families are, we released all those companies from any legal liability if the viruses hurt people or don't work. And they're not really approved, they're just kind of approved. Can you talk a little bit about that as areas of potential concern from somebody who's evaluating whether vaccine something they want to consider?
Sure. So in the past, the traditional way of getting medication into the FDA required several stages of clinical study. First, there was sort of a tolerance study in just a few adults to see how well they tolerated the medication. And then it would go to a phase 1 study where you started looking at side effects and efficacy, and then eventually to stage 3, 2, and 3 studies where you enrolled a large number of subjects to look at efficacy. In this case, because it was such an emergency, because so many people were getting infected so quickly, what the vaccine companies did was they did several of those stages simultaneously instead of back to back. So that dramatically reduced the amount of time it took to get the kind of data that you would normally get from a stage 3 study. So that kind of data was available within 6 to 8 months of when they started enrolling people into the study, instead of the years that it would have taken in the past. So that accounts for why it seemed to be such a short time. They didn't really cut corners. They simply did things simultaneously rather than end to end. But when the FDA made the decision to provide emergency use, the data that we're looking at was similar to the data that would come from a stage 3 study. And because it was such a national emergency, and because that data showed that the vaccine was both safe and efficacious, they provided that emergency use so that lives could be saved.
And what about, again, you talked about the package insert. One of the questions that came in at last week's talk that we didn't get a chance to talk about is, question reads, why is the package insert for each Pfizer COVID-19 vaccine blank? Is that informed consent? Are you familiar with that?
I have not looked at the package insert. It's difficult to me. I mean, the FDA has very specific regulations about the package insert, and no company would be allowed to put a blank package insert. The package insert that comes with the vaccine is not something that you would see at a vaccine clinic if you went to get vaccinated. It's something that the physician's office would see when they get the package delivered from the company, not something a patient would see. And those vaccine inserts, again, they're required, so they're required to describe every adverse event that occurs. And I don't know if you can see the vaccine insert online for COVID vaccine. It might be something you could Google and look for. But vaccine inserts are not blank. And what was the other part of that, Jeff?
Is that informed consent if you
No.
if it wasn't a
Okay. So whenever you do a clinical study is one that involves people, humans. When you do a clinical study, before you can start the clinical study, you have to submit the protocol, the design of the study. That includes the study design, all of the sheets that are required for collecting data, everything that's going on in the study. That has to be submitted to what's called a review committee, sometimes an internal independent review committee. This usually consists of a scientist, often a physician, usually an ethicist of some sort. It could be a minister of some kind, or even a philosopher. And they have to look at that study and determine if it's even ethical to do the study. Only if they give that approval is the study allowed to proceed. As part of that, anyone who is enrolled in a study is required to sign a letter of informed consent. In that letter, which also has to be approved by the review committee, explains to the subject what that study involves, what their expectations are, what their responsibilities are in terms of reporting data to the physician who enrolls them, and then allows them to become enrolled in the study. So everyone who is enrolled in a clinical study does have a letter of informed consent. Now to actually get a medication, you don't, I mean, when you get a prescription for antibiotics, you don't have to sign a letter of consent. You simply go to the drugstore and you get the medication and you take it. So taking medication doesn't require informed consent. Being in a clinical study does.
Okay, well, terrific. And then I could hear a comment about the packet insert. I'm going to have we'll post the link to Pfizer's packet insert online. Get what we've seen is that this is an evolving study, and so they're posting live data and updating that continuously. So I'm going to go ahead and send a link and get that posted. Another question that came in is just, do you think pharma has a really good track record? Isn't Pfizer one of the most highly litigated companies on the planet?
Well, let me tell you a related story. It'll give you kind of sense of this. Back in the early 90s, and I'm sorry, I can't remember which company it was, they came out with a vaccine for rotavirus. So rotavirus is a common virus that affects a lot of children, and it causes serious diarrhea. And this virus was called RotaShield. And it went through the normal approval process, was approved by the FDA, and went to market. And of course, when it went to market, all of a sudden there were millions of children who got the vaccine. And there were about half a dozen cases out of millions of children. There were about half a dozen infants who experienced what's called intestinal intussusception. In other words, the intestine kind of telescoped into itself, like a telescope folding up. and there were a couple of deaths. So the company immediately recalled the vaccine. Now, a friend of mine was at an international vaccine conference of virologists, and that's when that company made the announcement they were pulling that vaccine from the market. And they did that because they were afraid of litigation. They didn't want to be sued by people. And at that conference, physicians from developing countries and other parts of the world were absolutely outraged that they were pulling the vaccine. They said, I can't believe it. Because of a couple of spoiled kids in the United States, you're pulling the vaccine when we have thousands of infants who are dying every year from rotavirus. Why can't we have this vaccine? The benefits far outweigh the cost. So that gives you a sense of the kind of decision making going on in pharmaceutical companies when they weigh their best and risks and benefits of even putting a medication on the market. So they have to take things into account based on things like the very litigious society that we live in, over the benefits and risks.
What about the charts that you're looking at the wrong studies? That if you looked at the right studies, you'd see all the risks and all. And again, I've gotten a number of emails coming in, or not a number. I probably had 3 or 4 people reach out saying, "Well, I saw this one study and it looked concerning to me." And yeah, and here's a study that tells me that vaccines could never work. You're not looking at the right science. How do you evaluate what information sources to trust? How do you again, I think as a parent trying to make that decision for my child, I hear it in conversation from folks. I'm getting those emails. I see that on social media saying, I just saw this really alarming study out of India. I'm going to be a wait and see person. How do I?
I think the problem is that what some people are calling studies are simply stories. They're not really studies. When you say, I've seen a study, to be a study, that has to follow very specific guidelines. And you can judge the accuracy of a study, first of all, from where it's published. Is it a reputable journal? So what's a reputable journal? A reputable journal is a scientific journal that has a peer review process. When you submit a clinical study or even an animal study to a science journal, it goes through a process of peer review. In other words, other scientists who are experts in that particular field will look at that study and evaluate whether or not it is strong enough to be worthy of publication. The problem we're seeing now is that anybody can put together a story, and it can come from personal accounts or accounts that they've collected. So say someone goes onto a social media site and they collect all the stories they've heard from people who say they know someone who had, they know someone who, or of someone who died from COVID and vaccination. And they collect all those stories and they put a number on it. And then they write a paper saying, here's data that I collected on all the people who have been shown to die from COVID. And then that comes out as a study. That's not a study. In order to be confident that the source of your data is legitimate, you have to look at the source. When I see a story in the New York Times or even the Durango Herald about some health issue, I want to see where they got their information. It should have a reference, and you should be able to look at that reference. And that reference should take you to where the actual study was done. So you hear all these reports. I heard reports that so many people died from the Pfizer vaccine in their study. I went to the actual study that Pfizer submitted and was published in the New England Journal of Medicine that has the actual data. So to be confident, you have to know that you're looking at data that's based on fact, not on opinion. Data that's been very carefully collected and reviewed by experts before it gets published.
And I think, I think, again, this is not many people, but some of the pushback we've gotten from being part of a speaker series and hosting vaccine clinics is we've been duped by people with an agenda. The science isn't solid. If we're a science center, we should follow the science. How confident should we be in the science of the safety and effectiveness of vaccines? Is it, how clear and convincing is that evidence to you as you weigh both sides of that question?
All we have to do is look at the facts. The data are there. We now have actual data, actual facts on tens of millions of people worldwide who have been vaccinated. We have actual data, actual facts on tens of millions of people who are protected. And we can look at the track record. Remember all those vaccines that we now get routinely that I showed you before? How many people now die of measles or whooping cough? No one dies of smallpox. The track record is remarkable. I think a lot of the uncertainty people have with science is, they're looking at reports from the CDC and they say, oh, well, last week the CDC said this and now they're saying this. Science is a process of learning. It's not something that is set in stone. Science requires that you continue to evaluate information as it becomes available and then make changes as necessary based on new information. So if your original assumption was based on information that was incomplete, and you get more information so that you make a more informed decision and make newer recommendations, that's good science. That's making appropriate changes to improve the outcome. So when you see changes in recommendations, it's not because scientists are just willy-nilly changing their mind about something. It's because they continue to look at information, they continue to collect data, and they evaluate that data and then decide, oh, well, based on this new information, it's apparent that we should, for instance, add a booster, and that booster will add significant protection. And that data is collected worldwide. For instance, in Israel, they have been very effective in vaccinating a huge portion of the population and very efficient in collecting data, which has become readily available. So that's a good source of information. The other thing that's happened over the pandemic is that all the science journals and medical journals, normally if you want to download a clinical study from, say, the New England Journal of Medicine, it costs $35, $40 to do it. But since the beginning of the pandemic, all of these journals have offered free downloads to their papers so that information between and among scientists worldwide can be transmitted much more rapidly. I probably have over 100 papers in my virus file, and that's probably a fraction of what's been published. So you now have available actual data from multiple sources on dozens of clinical studies, as well as animal studies that led up to the production of these vaccines that allow you to keep up to date on the science. Science is not something that is embedded in concrete. It's a living, changing, it's science. It constantly evolves with new information.
What I know, yeah, knowing that you have that virus file, what I know about you that some listeners don't is just how much of your energy has gone to following the science on this. I've probably gotten more emails from you with studies and recommendations of what we should and shouldn't do here at the Science Center that I got from my wife in the last 2 years. So I do know how closely you've watched it. Have there been points where you had your mind changed? I think if one of the characteristics of science is being open to new information and adjusting based on evolving data, are there points in this last couple of years that you've had your mind changed?
Yes, actually. I mean, when the virus first hit, um, I was wearing a rag over my face as the mask. And then as I started getting more information, I realized, well, that's not going to work very well. So then I, started looking at mask studies to see what masks were actually effective. Um, and then I found one. It was a cloth mask, but it had a polyethylene insert, so it was actually 3 layers. So yeah, that changed my mind. And now more recent studies have come out saying, well, even the best cloth masks aren't as effective as N95 masks. So now I have an N95 mask. Um, so yes, I'm willing to learn and change my mind. Um, and it's important that another great example is with some of these fraudulent cures like ivermectin and these parasitic medications. Um, there actually have been clinical studies, and there also have been fraudulent studies that have been posted. So you have to learn to recognize what makes a study legitimate and what studies should be questioned. Um, and that's not to say that there haven't been studies published in science journals that have been pulled. One of the greatest examples is the study that linked vaccines to autism. That study was found to be fraudulent. Um, and the doctor who published that study, not only was that study pulled, but he had his license revoked for publishing. He made up data in that study. I personally have had the experience of working with a scientist who I did a clinical study with him, and I learned afterwards that he manufactured the data. And
well. I do appreciate you joining us tonight, and I'm sorry for the abrupt ending. We will do one more speaker series a week from today with San Juan Basin Public Health, and the recording of this video will get posted online. Can encourage you to continue sending questions to info@powsci.org, and we'll continue to do our best answering questions and trying to bring science to bear as people are trying to make well-informed decisions. Sure appreciate it. Thanks, everyone.